During digestion, complex carbohydrates are hydrolyzed into monosaccharides, primarily glucose. The catabolism of glucose is the primary source of energy for cellular production of ATP. The anabolism of glucose is also important in order to regulate blood glucose levels. The generation of ATP for use by cells involves 4 enzymatic pathways. These are glycolysis, formation of acetyl coenzyme A, the Krebs cycle, and the electron transport chain. Because glycolysis generates ATP in the absence of oxygen, it is considered an anaerobic pathway. To generate numerous ATP molecules, oxygen is needed. The remaining 3 pathways have varying concentrations of oxygen and are considered to be aerobic pathways. Glycolysis is the first step of glucose catabolism, and it occurs in the cytosol. The first part of glycolysis involves phosphorylating glucose and then breaking it into smaller phosphate molecules. The second part of glycolysis is the oxidation of the smaller phosphate molecules, thus producing reduced coenzymes and generation of ATP. As a result of glycolysis of a single glucose molecule, 2 ATP and 2 reduced coenzyme molecules are formed. Much more energy is still in the 2 pyruvic acid molecules that remain from the glucose molecule. Many more molecules of ATP can be generated from the energy still stored in the pyruvic acid molecules. However, this energy can only be harvested if the cell is well oxygenated. If there is inadequate oxygen, the pyruvic acid will be converted into lactic acid. However, if there is adequate oxygen, the pyruvic acid will be further harvested for much more energy in the mitochondrion. Cells that lack mitochondria generate significantly fewer ATP molecules. During the second step of glucose catabolism, formation of acetyl coenzyme A, each of the 2 pyruvic acid molecules resulting from glycolysis is oxidized to produce a reduced coenzyme. In addition, each pyruvic acid molecule loses a carbon dioxide molecule to form an acetyl group. Each acetyl group will bind with coenzyme A to form acetyl coenzyme A. The Krebs cycle takes place in the mitochondrion, as does the rest of aerobic glucose catabolism. During the Krebs cycle, the glucose molecule is completely oxidized. 4 more carbon dioxide molecules are released, for a total of 6. Two energy molecules are produced during the Krebs cycle. The remaining energy in the acetyl groups is transferred to reduced coenzymes. The primary function of the Krebs cycle is the production of many reduced coenzymes. It is in these molecules that most of the energy harvested from the original glucose molecule now resides. During the last step of aerobic glucose catabolism, the reduced coenzymes are used to generate ATP. This step occurs in the inner membrane of the mitochondrion. When the reduced coenzymes are oxidized, the energy is used to pump hydrogen ions across the inner mitochondrial membrane. Water is formed from this step, as the hydrogen atoms are released and combine with oxygen. Hydrogen ions are accumulated and form a concentration gradient between the inner membrane and the matrix of the mitochondrion. The concentration of hydrogen ions provides the force to produce ATP molecules. As the hydrogen ions diffuse down gradient across the membrane, they pass through a protein that synthesizes ATP. The process of producing ATP using the movement of hydrogen ions down their concentration gradient through ATP synthase is known as chemiosmosis. The goal of glucose catabolism is to harvest energy from fuel molecules to produce ATP. In turn, the energy in ATP is used by the cell to perform its work. Other products of glucose catabolism include the production of water and 6 carbon dioxide molecules. Glucose can be formed through 2 different anabolic reactions, glycogenolysis and gluconeogenesis. These reactions help to regulate blood glucose. Glycogenolysis is a process that breaks up glycogen and occurs in the liver or skeletal muscle cells. The hormones epinephrine and glucagon stimulate glycogenolysis. As the glycogen is hydrolyzed, blood glucose will increase and may be oxidized by the body cells to get energy. Through the process of glycogenesis, excessive blood glucose can be converted back to the polysaccharide glycogen for storage in liver and muscle cells. Glycogenesis is stimulated by the hormone insulin. As the glycogen is synthesized, blood glucose will decrease. Non-carbohydrate molecules, lactic acid, amino acids, and glycerol, can be converted into glucose in the process of gluconeogenesis. Like glycogenolysis, gluconeogenesis also occurs in the liver. The hormones glucagon and cortisol stimulate gluconeogenesis. As the glucose is synthesized, blood glucose will increase.